Hello everyone. Today we begins with the blood groups. Introduction. In the system of blood grouping, antigens are known as agglutinogens and antibodies are known as agglutinins. And the reaction between agglutinogen and agglutinins is known as agglutination. Blood grouping system. Major, minor and familiar blood grouping system. Major blood group system includes classical ABO blood group system and RH blood group system, whereas minor blood group system includes MNS blood grouping system and P blood grouping system. There are the few blood groups which runs within the families. These are the Kale, Duffy, Lutheran, Lewis, Diego and Kidd. So these all are the blood grouping system. But widely and majorly we are using this classical ABO blood grouping system and RH blood grouping system. ABO blood group system. On the surface of RBC, there are different type of precursor chains are present. These are the gene for A antigen, gene for B antigen and gene for AB antigen. But for the synthesis of either A antigen, B antigen or AB antigen, there must be the presence of H gene. H gene encodes the fucosyl transferase enzyme which transfers L-fucose to this precursor chain. So whole this substance is known as H substance. L-fucose plus precursor chain both is known as H substance. And the person who having the blood group O having that H substance on their surface of the RBC. Now we are going to see how the antigen A and antigen B synthesis takes place on the surface of RBC. Before the synthesis of antigen A or B, there must be the presence of H substance. H substance which encodes the enzyme fucosyl transferase which transfers L-fucose to the precursor chain. So this H substance is formed. Now the person who having the A gene which encodes N-acetyl galactosamine transferase enzyme which transfers N-acetyl galactosamine to the H substance. So this is our H substance. So whole this N-acetyl galactosamine plus L-fucose plus precursor chain are collectively known as antigen A or agglutinogen A. Alright. Now the person who having the B gene. But first of all there must be the synthesis of H substance which is due to the presence of H gene which encodes fucosyl transferase enzyme which transfers L-fucose to the precursor chain and it forms H substance. Now the person who having the B gene which is responsible for the activity of galactosyl transferase enzyme which transfers D-galactose to the H substance. So here the D-galactose plus L-fucose plus precursor chain collectively known as B antigen. Now the person who having the AB gene, first of all there must be the presence of H gene which is responsible for the fucosyl transferase enzyme which transfers L-fucose to the precursor chain and this forms the H substance. Now the person who having the AB gene, whereas the A gene is responsible for N-acetyl galactosamine transferase enzyme which transfers N-acetyl galactosamine to this H substance, and the B gene which is responsible for galactosyl transferase enzyme which transfers D-galactose to this H substance. So this forms the antigen A or agglutinogen A and this forms the agglutinogen B. So the person who having the blood group AB either positive or negative, they have on the surface of RBC both the antigens antigen A and antigen B. ABO blood grouping system. The person who having the antigen A on their surface of RBC, their blood group is A type. The person who having the antigen B on their surface of RBC, their blood group is B type. A person who having the antigen A and B both are present on their surface of RBC, their blood type is AB. And the person who neither have antigen A nor B on their surface of RBC, their blood type is O. ABO blood group system, which follows the Land-Steiner's law. This Land-Steiner's law is only applicable in the ABO blood group system. So here is the Land-Steiner's law. First law, if an agglutinogen is present on red blood cell membrane, the corresponding agglutinin must be absent in the plasma. Law number 2, if an agglutinogen is absent on red blood cell membrane, then corresponding agglutinin must be present in the plasma. What does it mean? If agglutinogen is present on the red blood cell membrane, for example, agglutinogen A is present on the surface of RBC, the corresponding agglutinin must be absent in the plasma. If someone having the blood group A, which having the blood group A which having the antigen A on their surface of RBC, if agglutinogen A is present on red blood cell membrane, then the corresponding agglutinin anti-A must be absent in their plasma. So here the agglutinogen A is present, so their corresponding agglutinin which is anti-A must be absent in their plasma. Anti-A which is absent in the plasma. If agglutinogen A is absent on their surface of RBC, then corresponding agglutinin must be present in the plasma. So here, if agglutinogen A is not present, then anti-A is present in their plasma. Alright. So blood group A. In the persons who having the blood group A, on the red blood cell membrane there is the agglutinogen A is present, but their corresponding agglutinin anti-A must be absent in their plasma. So here there is a presence of anti-B and antigen is A. In the blood group B, anti-A antibody is present and B antigen is present. The person who having the blood group AB, they don't have any type of antibodies in their plasma, but they having the presence of antigen A and antigen B on their surface of RBC. The person who having the blood group O, having the both antibody A as well as antibody B in their plasma, but they don't have any type of antigens on the surface of RBC. Clear? How is it genetically determined? So here in the columns you are seeing the ABO-LS inherited from the mother, and in the rows these are the ABO-LS inherited from the father. So that is the person having the blood group A, this person having the blood group AB, this person having the blood group A, this person having the blood group AB, this person having the blood group B, this person having the blood group B, this person having the blood group A, this person having the blood group B, and this person having the blood group O. Type O gene is almost functionless. Genotypes, phenotypes and percentage in India. O blood group is majorly seen out of all other blood group, which is around 32% in India. Later it comes to the A blood group, which is around 20%. Later it comes to the B blood group, which is around 40%. The last is the AB blood group, which is present in the 8% of the Indian population. A blood group is further divided into A1 and A2. Now RH blood group system. If RH antigen is present on the surface of RBC, their blood group is positive, and if Rh antigen is not present on the surface of RBC, their blood group is negative. So on the basis of this Rh blood group system, we are going to differentiate the person who having the positive blood group and the person who having the negative blood group. Inheritance. D antigen as a dominant gene. When capital D and small d, in this combination, the D antigen is the dominant gene, and when both the capital D are present, the person having the blood group Rh positive, and if the both the small d are present without any presence of dominant gene, then the person having the blood group Rh negative. Determination of blood groups. I already made one video over the blood grouping practicals of the hematology lab. Please go and watch this video. So we have three solutions: NTA, which is bluish in color, NTD, which is colorless, and NTB, which is a yellowish color. These are the artificially available antiserums, and this NTA solution contains antibodies against the agglutinogen A. This NTB solution contains antibodies against the agglutinogen B, and this NTD solution contains antibodies or agglutinins against agglutinogen D or Rh antigen. So we have three NTA, NTB and NTD solutions. Now the person who having the positive blood group, we are able to find the agglutination in NTA solution and in NTD solution. Remember this, whenever there is agglutination is present in NTD solution, the person having the blood group positive, and whenever the agglutination is not present on this NTD solution, the person having the blood group negative. The person who having the A negative blood group, we are able to find agglutination only in the NTA solution. If the person having blood group B positive, we are going to find agglutination in NTB solution and NTD solution. If the person having the B negative blood group, we are only able to find the agglutination in NTB solution. If person having the AB positive blood group, we are able to find agglutination in all NTA, NTB and NTD solution. If person having the blood group AB negative, we are going to find agglutination in NTA solution and NTB solution. If person having the O positive blood group, we are going to find agglutination in NTD solution only. And if person having the blood group O negative, we are not able to find agglutination in either NTA, NTB or NTD solution, because the O negative blood group does not having the any type of antigens, neither A antigen nor B antigen, and even not presence of Rh antigen. So we are not going to find any type of agglutination in NTA, NTB or NTD solution in O negative blood group person. Clinical applications of the blood grouping: in the blood transfusion, in preventing hemolytic disease, in paternity disputes, in medico-legal cases, in knowing susceptibility of various diseases. So that is the clinical applications of blood grouping, which is widely used nowadays in the clinical practice. So first one is the in blood transfusion, second one preventing hemolytic disease, third one paternity dispute, fourth one to solve some of the medical legal cases, and next or last one is the knowing susceptibility of various disease.